Good evening. Welcome again to the second part of the Cañete Valley. It is my pleasure to introduce John Forrest and Justus Barnes, who will both talk tonight about the changes, ecological and geographical, in the Cañete Valley. John, we very much look forward to your presentation. Just to let you know, there will be time at the end for John and Justus to take questions.
Okay, so as Paolo mentioned then, there was a first part to this talk. This is part two. I can recognise a few faces of people who were here for the first part, in which we introduced the Cañete Valley, and then the second talk was about the Bofedales, the high altitude marshy areas high in the Andes that we've been investigating. So this evening is two different talks, but also from the Cañete Valley. So the first two talks are on the Anglo-Peruvian Society website. So if you haven't had a chance to, you weren't here last year, then you can see them on the website. I'm going to talk this evening then about a project that was a result of the collaboration with the University of Cañete and Cavendish Laboratory at Cambridge that focuses on the river shrimp. I think most of you are aware that there's river shrimp in the Rio Cañete. And this was a project principally led by Noor, who is now back in Malaysia. And so I'm presenting on behalf of the team. So the talk this evening then is going to run through in kind of four sections. So first we'll say a little bit about the importance of the river shrimp in general in Peru, how they can be used as bioindicators, the significance of the Camarones of the Rio Cañete itself, and then the results of our studies. Okay, so if we go back over time, we can see that the Camarones on the coast of Peru, there's evidence that they've been of importance for quite a considerable amount of time. So in the evidence there in the ceramic vessels from the Moche, from the Nazca, and other cultures along the coast show that Camarones were an important part of the diet back then, and equally they're important part of the diet in Cañete today. And probably virtually all of you here who've been to Cañete, Lunawana, you probably did sample sopa seca de camarones or chupe de camarones. But it's something that is important for the people who live there. The river's divided up into a series of sections, and each one is kind of controlled, and there is a veda that runs January to March to try to prevent the harvesting of the Camarones in this period when the females are spawning, to try to safeguard the population in the long run. I mean, I think it's very much done by them. It's not something so, well, it says a ministerial resolution, so it is backed by the authorities, but enacted by these local commissions. So the Camarones then are found all along, or were found all along the coast of Peru in the valleys that bisect the coastal plain, and particular today, but today sadly that's no longer really the case. So particularly in the north of Peru, north of Lima, there's definitely been a reduction in the valleys that have evidence of Camarones. So for example, probably in the Patavilca there might still be some Camarones there. The Moche, I very much doubt that there's any Camarones, a population remaining in that river. I think in the Chicama Valley, in a tributary, the Rio Grande or Rio Grande, there's still a population, but generally in the north they've been heavily impacted by human activities, and the populations are much reduced or no longer there. Whereas in the south of Peru, there's still quite healthy populations, particularly in the far south and in the Cañete Valley. So taking, we could choose, we had to consider when we began this project what we would use as a bioindicator in the Cañete Valley. There's some different options, but really just monitoring water quality. You're only looking at water at that moment in time as it passes down the valley, and so it doesn't really give you a very good idea in the long run of water quality in the valley. Equally, if we took soil samples, perhaps in the vicinity of the valley, or on the valley floor, we would equally find that it just gives you a snapshot at that moment in time of that particular sample of soil in that location. And if we were to take some agricultural produce, then, well, that's absorbing some contaminants, but it's going to be harvested, they're going to be removed. It's not going to give a very clear picture of what's happening over a longer period of time in the valley with respect to the environmental conditions there. We wanted a species then that is going to give us particularly an idea of the water quality in the valley, and therefore we needed a species that's living in the river. And there's quite a lot of species of macroinvertebrates that we could study, but they're not all suitable. Some of them, such as those on the left, they're not going to survive in pollution of even low levels of pollution. So once the river becomes slightly polluted, they will disappear. So that's not a good species. And equally, on the right-hand side, the kind of species that we might find there, they're tolerant of very highly contaminated conditions. So again, they're going to survive right through until really the river is very heavily polluted. Whereas the shrimp can tolerate a certain degree of contamination, and it's a species that we're going to find over a significant distance within the channel, and is going to be a better indicator over a longer time period of what's happening in the valley. So the river shrimp is it's in the river, well, the Cañete River, and in the other rivers in the south of Peru it's fairly common. So we felt that we would be able to catch the number that we would need. You can easily identify it. You can't confuse it perhaps with another species. It's large enough to find and to catch. It's there all year round in the river. Well, we can't catch it in the La Veda in those months, January to March, but in the other months, potentially we could catch them. And they, as we've just said, they will tolerate reasonable levels of contamination and therefore give us an idea of what's happening to water quality in the river. We could then take the flesh of the shrimp and we could assess it for the trace metals that were accumulating within the flesh of the shrimp. So to do that, we would need a minimum of 300 grams of flesh of the shrimp. So in other words, the part you eat, when you're eating shrimp, we would need a minimum of 300 grams of it for the lab to process and assess what chemicals are within it. It might also tell us then what are the pathways, or we could investigate further the pathways leading to the build-up of the trace metals in the shrimp, if we were to find trace metals in the shrimp, to give us an indication of just how concentrated the metals are and perhaps what their origins are. So the river shrimp therefore presented itself as a kind of ideal candidate to study. There is only one genus of river shrimp on the west flank of the Andes, from southern Chile up into Peru. The shaded area on the map doesn't quite indicate the full extent of the river shrimp within Peru. In fact, it's not a very good map at all. But unlike other countries, there is just a single species. So that also would make things easier. So the river shrimp then, if we begin over here then, we've got the mature river shrimp, the males with their bigger claw, and the females on the left there, living up the river. They've migrated up there as juveniles, or the females have returned there, because the males, once they reach a higher point in the river, they stay there. They don't then return down. Mating takes place in the higher part of their territory, and then it is the females that make the journey back down to the coast, to the brackish estuarine lagoons or the mouth of the river. So it does seem that they need conditions that are slightly saline, because here the seawater is penetrating through into these lagoons to spawn. So the females spawn in these lagoons down by the mouth, and then the young develop, and the females and the young then migrate back up the river, back up to these higher areas where they then stay, and the cycle then repeats itself, with the females going back down the river and the males staying in the higher altitudes. As you can see there, they do travel up to quite a significant altitude, approaching 1,500 meters upstream. So the River Cañete then, Cañete is here, Imperial, Lunawana, the town here. And so the Camarones then are found all the way up to currently this kind of area around Cotahuasi. It's possible that they're going up the Lincha Valley, which is this one, to a site somewhere about here, and maybe a bit further up above Cotahuasi. And at one point they went up to just above Capayucas, but we'll mention later why that's no longer the case. So they are migrating quite a significant distance further upstream from Lunawana, and then the females have to come all the way back down to those lagoons near the mouth. As you can see, that's a distance of about 80 kilometers that they're making each year to spawn. I say each year, perhaps I'm not certain that they would necessarily do it each year, but we assume that they might be doing it each year, or at least once in their lifetime. So we had to set about catching some shrimp, and we thought maybe we'd be able to do it ourselves. And we put on our wellies, we go down to the river, and we turn over a few stones, and there would be the shrimp, and we kind of scoop them up, and it would be very straightforward. But definitely that was not the case, and we quickly discovered that it was impossible to catch even one shrimp, and we needed a professional. And so we encountered this chap, Lely, who was a local and belonged to one of these commissions. And sometimes they say that people are, they kind of reflect the jobs that they do. Well, Lely, he was the perfect physique for catching shrimps, short legs, a stocky body, and quite long arms. And he was ideally suited to catching the shrimp. But unfortunately, a bit like the shrimp, he was a bit hard to find and to pin down to assist us. So we only used him the first time, but he had no problem at all in scurrying across the riverbed and catching the shrimp in no time. But as a result of his efforts, we discovered that actually catching the shrimp in the daytime was not the time to do it. And that really it is dusk that is the time to catch the shrimp. Because the way they do it is to have a lantern, and they then can see the eyes of the shrimp, and if they see the eyes of the shrimp, then they can catch the shrimp. Whereas in the daytime, it's much harder to spot the shrimp. So thereafter, catching the shrimp turned into an activity between about 6 and 10 in the evening, and they would go into the water, and we would say goodbye to them and hope that 2 or 3 hours later they would emerge with our shrimp. But these guys are, they do it all the time, so we were confident. They had the gear for it, so we were confident that they would be able to come up with the shrimp, the quantities that we needed. And so there are the two main people who we used. Franco on the right, who stepped in when we were talking in the breakfast in the hotel one morning, saying, we haven't got anybody, Lely's not shown up, what on earth are we going to do? And he said, well, so he was managing the hotel. He said, well, I can do it. I have a license, I'm allowed to do it. I've got all the gear. So thereafter, he became the guy to collect the shrimp. Now, the shrimp, in terms of catching males such as this one, was quite straightforward, because we might, to get 300 grams of flesh, you might only have to catch 15 shrimp. They were a bit harder to find, but that meant that was relatively easy. Whereas for the females, which are far smaller, as you will see later, they perhaps had to catch 100 to produce 300 grams of flesh to analyse. Working at night meant that we then had to have coolers with ice, and we put them on ice overnight in the coolers, and then early next morning we would begin the task of processing them. So that was rather than processing them at the time in situ, because on the first attempt this was not the ideal conditions in the desert to undertake the task of extracting the flesh of the shrimp or the hepatopancreas for analysis there. So we had a lab with the University of Cañete in Lunawana. We had our own laboratory space where we could set up and undertake the whole process early the next morning. So every shrimp we photographed, we measured every shrimp that we were using, and then extracted the flesh that you can see accumulating there, ready for analysis at a lab in Lima. So students, lots of students from the university took part. So it was good experience for them to do something practical, to go out into the field and to work in the laboratory. So it wasn't just theoretical things that they experienced much of the time, but it was actually actual research and hands-on activities. So our findings then are divided into 6 sections then. So the biometric data, something about the taxonomy of the species, distribution of them, their diet, bioaccumulation of trace metals that we found within them, and the possible sources of those trace metals. So measuring all the shrimp then, the data that resulted, it doesn't say what the size, the population size was there, which is maybe an omission, but we basically measured all the shrimp that we caught and which we extracted the flesh from. So the males were significantly larger than the females, so almost more than 50% larger. And the largest male was maybe this one at over 25 centimeters. So, you know, quite a lot of the males were over 20 centimeters. So quite a significant size. And as I said before, they were all from the upper areas. If we caught any males lower down, then they were much smaller because they were juveniles and hadn't yet grown larger. Some of these ones might be several years old. Again, not possible to determine exactly how old. There's also an interesting trend if you look at the dates there, that of course this was interrupted by COVID and we had to have a whole year out, and it could be that in that time there was less fishing or catching of Camerones in the river, and so you noticed that the males in particular increased in size, but the females as well were larger in size, maybe because they were not caught in the time of COVID and they had time to get a bit larger. In terms of weight, the largest was approaching 100 grams, and again the males very significantly larger, 4 times larger than the females. So in terms of weight and length, so you know, as we undertook the study, in most sites are 3 main sites and an additional 4th site. So the 4th site was the site highest up the valley, and you can see that when we were there we found no females, only males, which was perhaps to be expected with all the females having migrated back down to these lower sites. And then the lowest site, the lagoon, we found no males. So it kind of reflects the migratory process involved. In the first visit, we took samples and sent it to a laboratory in Lima for DNA analysis, just to confirm, just to prove the species, because very few studies, or no studies. I don't think there's any published study of the shrimp in the River Cañete. So this is the first, perhaps people have done some other smaller studies, but this was the first kind of larger scale study of the Camerones there. And so the laboratory in Lima received its sample of 300 grams of flesh, and they were able to confirm the DNA of the species, to confirm that it was the Andean river shrimp. I mean, the other valleys along the west coast of Peru, we assume that they're all the same, but possibly there will be some variation between them, as they appear to be certainly between the north and the south, more isolated populations, isolated to a degree from each other. So the 4 sites that we had, as I said before, went up to nearly 1,500 meters. So we were able to confirm that there were populations up to that level, more or less where we found where the populations exist. So the territory or the extent of the populations, that there were the differences between the males and females, more males upstream than downstream, and that there is clearly a migratory pattern taking place within the river system. So we also analysed the shrimp for their diet to try to establish what they're eating, and it seemed that the majority of their diet then is plant-based, but also evidence that they're consuming macroinvertebrates. So a further analysis could be undertaken to investigate that, to investigate exactly what they're eating, and that might be important if we're trying to establish the origin of any trace metals that are found within them, to go back through the food chain of what they're eating to how the trace metals accumulate in the shrimp. So the trace metal in accumulation, so we've got here then in black arsenic, copper, iron, selenium and zinc. So we can see that there is, so this time it's going this way towards the mouth of the river. So we can see there is a slight accumulation then of these trace metals as we go downstream between the sites. So sites 10 to 12 here was the highest site, but the main upper site was that one, which was located just above Lunawana, the one just below Lunawana, and the one at the mouth. So a slight accumulation of those trace metals and some evidence of trace metals accumulating within the flesh of the shrimp. However, the analysis shows that whether it's arsenic or copper, zinc, that the level of accumulation is below the tolerance levels that are considered to be of significance. So there clearly is an accumulation. Sometimes the level was greater in the shrimp than it was in the water, because we're also analysing the water for these metals as well. But it's always below the level that internationally is set as a level of concern. So that means, the good news is that means eating the shrimp is okay, but perhaps as long as you don't eat them every day for lunch and dinner. So as I said before, further analysis could help to show perhaps where those trace metals, how they're accumulating, because they're probably not picking up the trace metals directly from the water. It's probably more likely through their diet. So to understand that a bit better, one would have to go a step back down the food chain. So there is evidence there to suggest as to how that might come about. So whether it's throwing rubbish away, the use of pesticides. Not that the analysis of pesticides in the water showed a very significant accumulation of pesticides in the water in the lower Rio Cañete, but clearly, evidence of discarded materials from the use of a variety of chemicals. In some cases, some sewage perhaps entering the river. But in the main part, the lower Cañete doesn't have large populations living along it that discharge into the river. Imperial and Cañete San Vicente, their effluent is taken away in drainage canals further north and doesn't enter the river, which obviously is good from many points of view, but particularly from the point of view of the shrimp there. So there are possibilities as to how the trace metals are entering there, but that would somebody would have to undertake a further study to investigate a bit more exactly the food chain that's happening there. Of more concern for the shrimp, those dependent on the shrimp in the valley. So the valley is divided into 4 commissions, one in the lower, 2 in the middle, and 1 in the upper, and they control the river. They say there's about 35 families that depend on shrimp for their livelihood, but there's many other people, such as our guys, who are able to take shrimp out as and when, but they probably have other jobs and they're not taking shrimp every day. But it is therefore of some significance to the local communities, and of course not just the people taking the shrimp from the river, but all the restaurants in Lunawana and on the road leading into Lunawana that are serving shrimp are also semi-dependent on the shrimp. But the commissions, we had to go to the commissions, we had to meet the president, we had to seek permission to take the shrimp. We couldn't just go to the river and with our guys and take the shrimp out. We had to go and get permission each time that we were there. But the more significant thing for them is the Capillucas Dam, which is just upriver from Cotahuasi, and it's suggested that the shrimp used to inhabit the river beyond the reservoir here, perhaps up to over 1,500 meters in the valley, But of course that's been cut off now, and for much of the year the amount of water in the valley below the dam is just a trickle, because the water's taken by pipe through the mountain and comes out much lower down, just above Zuniga. So the shrimp are having to cope with that, a reduced level of water in the river, but more significantly is the fact that every so often they cleanse the dam, and that means that they let out a lot of additional water from the reservoir and flush it out down the river, and it is the sediment that is the problem for the shrimp, that they just can't cope with the quantity of sediment that is then released in one go, and unless they can escape somewhere hidden, we've seen evidence that that is likely to kill a significant number of shrimp, significant quantity of shrimp. On the odd occasion this happens. Now the authorities there do give a warning that they're going to do it, but it's not much good to the shrimp to know that this vast quantity of dirty water is coming down the river, and that's what concerns the local people the most, that happens. Okay, so in summary then we can say that we know something about the distribution, the population, perhaps some idea of the health of the population, something about the diet of the shrimp. We know that they do accumulate some trace metals, but currently it's not at a critical level. We have an idea of what those trace metals are, and that we know that the accumulation, as expected, is greater lower down the valley. So definitely the Andean shrimp was a good choice. It's a good indicator of contamination in the river there. I mean, they are, like that, they are beautiful creatures, and they're tasty creatures as well. And we'd all like, I think, to think that a healthy population remains there. Our studies came to an end there, but there's plenty of scope for further studies there. Now, the unfortunate thing is that Peruvian universities, such as the University of Cañete, are very restricted to their careers that they offer, and they don't. Although they offer agriculture, that doesn't include shrimp. So it includes agriculture in terms of farming. So really, there's nobody there who has a particular interest in it, and they don't really have a way, even through the kind of the tourism side of things, wouldn't be expanding or developing this kind of study. Another challenge is that the staff there is about a quarter of the staff are permanent, but about 3/4 are contracted each year. So they come in just for 9 months or so. So they're very interested when they're there, and they would come out with us, but they're not going to be there the year after and the year after that, such as like us, to develop a project. And whether they're contracted staff or permanent staff, they almost certainly, none of them are from Cañete or live in Cañete. So they've all come from somewhere else, or they all have homes in Lima. So each weekend and holiday, they go back to those places. So it was a challenge to find some staff there who were able to kind of fully participate with us in the study, and perhaps would be interested in carrying on with it. Also another challenge was that while we were there, we had 2 changes of president at the university in the 4 years, and 4 changes of vice president of investigation. So each time we were working with somebody new, needing to explain the project and so on. So that is another challenge, when you're trying to work with a Peruvian university on a project along these lines. Some universities probably go to Peru perhaps and are able to work more independently, and if anybody could take part with them, that would be good. But the idea of this project was to do something more integrated, and to really try to stimulate, encourage and assist the University of Cañete to develop its environmental research. So just ending on that point about some of the challenges then with this kind of project. Okay, thank you. I think you can just see if your one comes up. You want a quick question now, or do you want to just go straight to the next one?
Do you do exchange with students from
Some students did come here twice from Cagnette. They didn't come directly within the project, but they came because of the project. So the project meant that they could visit Cambridge. Without the project, they wouldn't have been able to. And they were students of tourism, accountancy, administration, farming. So obviously only one of those directly relates to the project. But because of the project and our experiences, we were able to offer something, you know, despite our academic backgrounds, we were still able within Cambridge to organise activities and so on within tourism, accountancy and administration for those students to make it a more kind of comprehensive engagement with the University of Cagnette. Because that is also another thing with the universities there, that perhaps they see it that, you know, we'll just work with a very small group of staff and students, and the rest of them are thinking, well, you know, what's in it for us? You know, how are we going to benefit? Quite a few staff, some staff came from different sectors, so more from the kind of IT or communication side of things. But the project was only set up for staff connected to the project to come, but because there was some other funding that they secured as a result of the efforts of one particular teacher, she twice brought groups of, I think, 12 and 15 students over. So other students had the experience of visiting the UK and did, it wasn't just a passantia of, you know, relaxed tourism visits. They actually had to do some work related to their studies.
Yeah, they're very keen on coming. They're not so keen on spending hours in the laboratory, but you know, they are very keen on sending students and staff.
But the students who came had to do a proper kind of investigation in their subject area when they were with us.
Cagnette is the third project. So previously we've been in Moquegua and then Barranca, now Cagnette. And now we're somewhere else. Slightly from.
Where are you now?
We're in Huanta.
Oh, Huanta, Ayacucho.
Yeah, Cucho, yeah.
So next year, if you're interested. But,
it's a very different kind of project.
Sorry, given the difference in size, when we have our troupe, do we just eat males or do they catch
Mostly males, yeah.
females as well?
more the males,
Yeah.
I would say, but also both. I would say you would find. I mean, definitely if you have one presented on top, or as the larger ones would be the male. So anything over about that, that's probably, what was it? The female was 10 centimeters or so maximum. So many are smaller.
Yeah,
So they'll be within the dish maybe, and on top will be the. I'm afraid the big male. Yeah.
It is you didn't say, but it is actually a threatened species. And you know, it has disappeared from most rivers in Peru. The Pativilca, which used to have a similar sort of economy to the Cagnette, now doesn't have any. All the restaurants that used to sell chupe de camarones now can't. And that's because of mining in the Cordillera Huayhuash.
Probably a coal mine there.
coal
It's just
mine,
putting
yeah.
too much sediment into the river.
No voy a preguntar en español.
Ah.
¿Había diferencia entre el male y el female en cuanto a contaminación?
I would say that off the top of my head, I haven't got the data for that, but the females, because they go lower down the river and there would appear to be more contamination lower down, I think we found a greater accumulation of the trace metals in the females and less so in the males because they're living above Lunawana. I mean, there's no mines really directly affecting the Rio Cagnette. There's no significant agriculture higher up. There's hardly any urban areas, so other kind of waste there isn't. So the males have got it pretty good living up there. And it's the females who have to make the huge journey, take the contamination, get scooped up for the restaurants, and and go back up again.
Yeah.
Yeah.
Yeah. And
Okay,
I mean,
shall we?
Yeah.
You got the thing, yeah.
Yeah, I have.